Hi, so this wee video is about the synthesis and transport of proteins from the Advanced Higher Biology course, Unit 1, which is cells and protein, and it's from Key Area 2, which is about proteins, and this is Part B from that key area. So what we're trying to do is learn about the importance of intracellular membranes in eukaryotes, the protein and lipid synthesis that occurs in the endoplasmic reticulum, and also the transport of proteins. We'll be looking at stuff like the Golgi apparatus, lysosomes, the transmembrane protein signal sequence, which is important, and post-translational modification that occurs in the Golgi apparatus. Now, key players in protein synthesis and transportation. So from S1 to S2 up to higher, you will have looked at the cell structures of a cell. We have not mentioned two important ones, endoplasmic reticulum and the Golgi apparatus. We'll be looking at both of those just now. First, we're going to actually learn a bit about the actual key players before we actually learn about their roles. Now, the endoplasmic reticulum, also known as ER, is a transport system of the cell. It's basically a network of tubes that are really, really close to the nucleus, and they're actually joined to the membrane around the nucleus. Okay. There are two types of endoplasmic reticulum. Looking at the light blue type just now, and this is the rough endoplasmic reticulum, often called RER, and it's actually got ribosomes attached to it. If you remember, ribosomes are the site of protein synthesis. There's free ribosomes that float freely in the cytoplasm, and there's also some that are attached to the endoplasmic reticulum. And they produce proteins that are to be transported. Now we've also got the smooth endoplasmic reticulum, which is SER. It's got no ribosomes, however it does produce enzymes and lipids. Okay, so it's not just proteins that we're talking about here, we're talking about lipids, which are a type of fat, and those are going to be transported as well. So the ER transports the proteins to the Golgi apparatus, and that is responsible for actually packaging the proteins. It's a bit like a little post office. Now the Golgi apparatus is basically a series of discs, like membrane discs, so little flattened membrane discs that are piled on top of each other, so layers and layers of membrane. Now if we just, I'll just grab a pen, if we look here, you've got one disc there, which then piled up on another disc, piled up on another disc, okay? And basically the Golgi apparatus receives the packages of proteins and lipids from the ER, and then it sends these packages to lysosomes, which we'll look at in a minute, the plasma membrane, so like the cell membrane, and to outside of the cell. Okay, so there's lots of different places that these packages of proteins and lipids can go to. Now, the lysosome is a structure whose primary role is to digest big molecules. Now, these big molecules could be cell waste, maybe like dead bits of cell. It could be things that are meant to be there, like pathogens or microorganisms, or any sort of waste products. Now, the lysosome is basically a membrane-bound organelle containing hydrolases. And a hydrolase is basically a type of enzyme. Okay, now these enzymes are responsible for just digestion. Okay, as I said, any of the big molecules. So they are the cleanups of the cell. They can digest anything from proteins to lipids to nucleic acids to carbohydrates. They can digest anything. Now, in order for the Golgi apparatus to get its packages of proteins and lipids to the lysosome or the cell membrane or whatever structure it's sending it to, it needs something to travel in, and its vesicles are basically what they travel in. They are little membrane-bound compartments. Now, if we have a little look here, just look at this one just now, you can actually see that it's got a phospholipid bilayer, just like any other sort of membrane-bound vesicle. And basically what's inside the vesicle will be the protein or the lipid that the Golgi apparatus has finished packaging. Now if you look at the other diagram, this is an example of a vesicle that is actually moving towards the cell membrane to release a protein outside of the cell. In actual fact, upon looking at the diagram, it's actually going the other direction, but it works the same way. If the arrows were just pointing this way, it would be showing the vesicle moving towards to joining the membrane and then it opening up here to release the contents to the outside of the cell. So we're just going to look in more detail at how we actually produce the lipids and proteins in the endoplasmic reticulum. So just a little recap on this diagram here. We've got our nucleus, we've got our rough endoplasmic reticulum with the ribosomes attached, and then we've got our smooth endoplasmic reticulum. So the ER in more detail. This is where our lipids and proteins are produced. Our rough ER has got the ribosomes on the cytosolic face, which is basically the edge, the membrane of the ER that's facing the cytoplasm. Okay, so it's like the outside of the ER. While the smooth ER, it lacks ribosomes altogether. The smooth ER is responsible for lipids, producing the lipids, and what it does with these is inserts them into their membrane, and this will allow them to be released for travel. Now, the cytosolic ribosomes, so we've got some ribosomes that are actually on the end of the endoplasmic reticulum, and there's other ribosomes that are basically free floating in the cytoplasm. If we look at this diagram here, we've got our cytosolic ribosomes there, and here we've got our ribosomes are attached to the ER. Now, protein synthesis, whatever protein we're talking about, begins in the cytosolic ribosome. Doesn't start in the ribosomes at the ER, it begins in the cytosolic ribosome. Okay. Now, depending on the protein it's producing, or the ribosome is producing, it might go to the endoplasmic reticulum, or it might not. So, cytosolic proteins, so proteins that are going to stay in the cytoplasm, complete their synthesis in the cytosolic ribosome. However, proteins that are going to be transmembrane proteins, so proteins that are actually in the membranes of structures, they complete their synthesis in the ER. So, what happens is these transmembrane proteins, as they are being translated, they actually carry a signal sequence that halts translation and directs the ribosome to dock, basically latch on to the ER, making that ER become a rough endoplasmic reticulum. It now becomes RER. As soon as that ribosome attaches onto the ER, it is now rough endoplasmic reticulum. Now, this signal sequence is just a short strand of amino acids at one end of the polypeptide chain, and this is what determines the actual location of the protein in the cell. No, sorry. Now, after our ribosome has docked onto the ER, translation then continues and the protein is inserted in the membrane of the ER. And once it's inserted in the membrane of the ER, that's when it can actually travel to where it's actually required. Now, we're just going to go over the synthesis of the membrane component of the RER. Right, so stage 1, protein synthesis occurs in the ribosome that's floating in the cytoplasm. Okay. Then, if we look here, we've got an SRP, which is a signal protein that's actually been released from the membrane of the ER, and it comes and binds to the signal sequence, which stops translation at that point. This SRP that's bound to the receptor directs the ribosome to attach to the ER, forming the RER. Okay. Translation then restarts, and the protein is now inserted into the membrane of the RER. Of the RER. So what we've got here is we've got our ribosome, we've got the translated bit of messenger RNA, and it is attached to a station on the RER. Okay. When that protein is finished being translated, the ribosome will then detach itself again, leaving the protein in the actual membrane of the ER. So at this point, we've produced proteins that are free-floating in the cytoplasm. We have also produced proteins that are going to be transmembrane proteins, and we've produced lipids. We've not actually packaged any of these yet, so we've only just produced them. We're still in the endoplasmic reticulum. Now, it's at this point I'd normally pause, and the pupils in the class might wish to take notes. So I'm just going to summarise what we've talked about so far. And if you want to pause the slides at any time, take notes, do so. Okay. Right, so to summarise our synthesis of membrane components, lipids and proteins are both synthesised in the ER. Our rough ER has ribosomes on its cytosolic face, so facing into the cytoplasm, while the smooth ER has no ribosomes. Now the smooth ER is responsible for synthesizing the lipids, and once those lipids are produced, they're then inserted into the smooth ER's membrane. Now all proteins, whether they're transmembrane proteins or ones that will just be released free into the cytoplasm, they all start their production in the ribosomes that are free floating in the cytoplasm. Now the synthesis of cytosolic proteins is completed there, and then they remain the cytosol, the cytoplasm. And transmembrane proteins, those are the ones where the ribosomes end up moving to the ER. The transmembrane proteins carry the signal sequence that halts the translation, moves the ribosome that's synthesizing that protein to dock onto the ER, and then at that point, translation is continues and then completes, and that protein is actually inserted into the membrane of the ER. Now, so what we've got now is we've had the ER and the ribosomes producing the proteins and the lipids, and now we're going to look at how they can actually move from the ER to the Golgi apparatus for packaging. Right, when the proteins are in the RER, they're transported by vesicles and they bud off from the ER. Now if we look here we've got our nucleus there. We've got our rough endoplasmic reticulum here and our smooth endoplasmic reticulum there. And what you can see is a vesicle here forming and budding off and moving towards the Golgi apparatus. When it gets the Golgi apparatus, the vesicle fuses with the membrane of the Golgi apparatus and releases its contents into the Golgi apparatus. Okay. Now, as the proteins move through the Golgi apparatus, remember the Golgi apparatus packages the proteins, okay? So, and this is called post-translational modification. The Golgi apparatus will actually alter the protein a little bit. It might add on a hydroxyl group. It might cut a little bit off. The most common one is the addition of a carbohydrate group. That's the most major modification. This diagram shows how we can get one protein, and the Golgi apparatus can add various components or molecules to change that protein ever so slightly to give it a new structure and therefore a new function. So the molecules have to also move through the Golgi apparatus. Now if you remember, the Golgi apparatus is a series of discs, membrane discs that are kind of piled on top of each other. Now, you remember that the endoplasmic reticulum released a vesicle with the proteins into it. It then merged to the membrane of the Golgi apparatus and released its contents inside the Golgi apparatus. But it's now got to move from one Golgi disc to the next. So what happens is it's going to move through the Golgi apparatus. Then when it gets to the end of a disc, a new vesicle will bud off here and then travel to the next disc. Then it'll fuse with the membrane of that disc and release the contents inside it. Remember, the whole purpose of our molecules traveling through these Golgi discs is post-translational modification. So as the proteins move through the discs, enzymes will be catalyzing reactions to change the actual structural formation of that protein. Okay. For example, enzymes catalyze the addition of various different sugars at various steps of the Golgi apparatus to form these different carbohydrates. Remember, the addition of a carbohydrate group is the most common post-translational modification that occurs. So once the protein's moved to the Golgi apparatus, it's been modified in all the ways it's meant to be modified. It then is complete and needs to go somewhere. So in order for it to travel to where it's meant to go, be it the plasma membrane for being released outside the cell, or maybe to the lysosome or another structure, it's going to form yet another vesicle at the membrane of the Golgi apparatus, so it'll bud off, and then that vesicle will move to the plasma membrane or the lysosome. Now, we've talked a lot about vesicles moving between membranes, so be it from the ER to the Golgi apparatus, or vesicles moving in between the Golgi discs, or maybe the vesicle moving from the Golgi apparatus to the lysosome or the plasma membrane. We're now going to look at how those vesicles actually move. Now, they move along microtubules. These microtubules are long tubes, basically, purely for traveling. Now, they don't just attach by themselves. There's actually a motor protein that is attached to the microtubule and will then hold on to the vesicle and then just carry it along the microtubule. A little bit like a conveyor belt, or maybe an Arctic lorry truck driving along roads taking deliveries to supermarkets. Our motor protein is driving along the microtubules, taking our vesicle from the ER to the Golgi apparatus. So what we've got now is we've got our proteins that were made in the ER. We have moved them to the Golgi apparatus. We have modified them. And then we have moved our proteins to wherever they're meant to go along the microtubules by our vesicles. Now again, we're just going to summarise what we've just talked about. So if you do want to take notes, just pause. These are the types of notes that the pupils in my classes would be taking. Right. Once the proteins are in the ER, because we've already done that, but we've already made them, they're transported by vesicles to the Golgi apparatus. Remember, the vesicles bud off from the ER, and then they move by the microtubules, move along the microtubules, and fuse with the Golgi apparatus. These molecules, the proteins, are going to move through the Golgi apparatus. Remember, the Golgi apparatus has various discs piled on top of each other. They're going to move through these discs by the same manner of vesicles budding off each disc to fuse on to the next one. Whilst the protein moves through the Golgi apparatus, they undergo post-translational modification. The most common one is the addition of a carbohydrate. Enzymes catalyze this addition of the carbohydrate, and they'll catalyze by adding sugars on top of each other to make more and more complex carbohydrates. Vesicles that then leave the Golgi apparatus, remember, will bud off from that membrane, will take the proteins to the plasma membrane, maybe for releasing outside of the cell, or to the lysosome. Now remember, the lysosome was the structure that is the big cleanup structure of the cell. It's filled with lots of enzymes that can destroy big macromolecules. Again, for these vesicles to actually move from ER to Golgi apparatus, through the Golgi apparatus, from the Golgi apparatus, the plasma membrane and the lysosome, it's going to move along our microtubules, being carried by our motor proteins. And then it will fuse with whatever membrane, maybe of another structure, if that's where the protein is going. The vesicle will then move to the membrane of that structure and fuse with them. Or it might go and fuse with the actual membrane of the cell, ready to be released out into outside of the cell. Now we've been talking about transmembrane proteins and secreted proteins. Now secreted proteins are the ones that are destined to be released into the cell. The transmembrane proteins are the ones that are destined to be stationed in a membrane somewhere be it the cell membrane or it could be a membrane around an organelle. It all starts off the same, going from the nucleus to the ribosome to the ER to a vesicle to the Golgi apparatus for post-translational modification, then another vesicle, and then that's where it's different, okay? The transmembrane vesicles are going to go to the membrane that the protein is meant to be stationed at, and the vesicles for the secreted proteins are going to go to the cell membrane, where there's actually going to be another post-translational modification to actually make the protein work, and that's what we're going to be looking at. Right, so the secretory pathway. Secreted proteins are translated in the ribosomes, we know this, on the RER, we know this, and then it enters the lumen of the RER, and the lumen, in actual fact, is the insides of the RER. Okay, that's what we see here. Here's the outside of the RER, the membrane outside the RER, and what happens is our proteins that are formed then get released into our lumen. Okay. Examples of secreted proteins are like peptide hormones, digestive enzymes, basically any protein that's going to be traveling around the body. So just as we talked about before, these secreted proteins are going to move along the Golgi apparatus and are packaged into vesicles. But the secreted proteins go into things called secretory vesicles. Okay. Now, these secretory vesicles move and fuse with the plasma membrane as described before, releasing the proteins out of the cell. This isn't new information yet. This is the new bit. So, when our secreted proteins are released into the outside of the cell, they're actually not active. They're secreted as inactive precursors. They're not in a functional state. Now, this is to avoid proteins being released from the cell and then doing jobs that aren't actually required. Okay? By being released in an inactive precursor, it means the protein will later on get turned on when it's actually needed. It's going to get put into its functional state. Now, this process of being transformed into a functional state, it's another example of a post-translational modification. It's called proteolytic cleavage. Okay. Now, what happens is it's when you get one bit, you've got your big protein chain, and then one bit of the protein is going to bend round, form bonds with the other strand, the other section of the protein, and then just cut bits of the protein off and chuck them out. So what we've got here, if you look at A, we've got one big long protein. B is the process of our proteolytic cleavage, and you can see that there's going to be some bonds that are being made here. Okay. And then the final stage is to then cut off the bits of the protein you don't need. So look, this whole pink section is no longer present in our active protein. And after the proteolytic cleavage, our protein is actually active and can function and carry out the jobs it's meant to do. As I said, the proteolytic cleavage is all about making and breaking bonds. It's a process of breaking peptide bonds between various amino acids in the protein to cut bits off, which is what we're seeing here. Now, digestive enzymes and insulin are both examples of secreted proteins that require proteolytic cleavage to become active. We're going to look at insulin first of all. Now, a precursor to mature insulin is pre-proinsulin. Okay, and you can see it's this long one here. Now, we've got one round of proteolytic cleavage to turn it into proinsulin. And you can see here that the signal sequence is cut off and we're getting some bonds starting to make here in between these sections there, these amino acids. We then have another round of proteolytic cleavage where we've got a whole strand, this bit here gets cut off and removed. And that is what produces our mature insulin. And this is the insulin that's actually going to carry out the function of removing excess sugar from the bloodstream. Now, this process of proteolytic cleavage means that we can have the protein, pre-proinsulin and proinsulin, floating around, not doing anything to the blood sugar in the bloodstream. And then as soon as we're actually requiring the removal of sugar from the bloodstream, that's when our proteolytic cleavage occurs so that we can get our functioning mature insulin. I'm just going to summarise what we've talked about in terms of the secretory pathway. As before, feel free to pause and take notes if you need to. Right, secretory pathway. Secreted proteins are translated in the ribosomes, just like everywhere else, on the RER, and then they enter the lumen. Peptide hormones and digestive enzymes are examples. The proteins are going to move through the Golgi apparatus, and then they're packaged into secretory vesicles. These vesicles move to and fuse with the plasma membrane, releasing the proteins out of the cell. Often these proteins are inactive. They're inactive precursors, and they require proteolytic cleavage to produce active proteins. Proteolytic cleavage is just another type of post-translational modification. Digestive enzymes are one example of secreted proteins that require proteolytic cleavage to become active. We looked at insulin as an example. That was a long video. It's a big key area. Hopefully that's helped clear some stuff up and explain things about the synthesis and transport of proteins. Okay, thanks guys, I'll see you soon.